Okay, let's unpack this. Today, we are undertaking, well, a really intensive deep dive into the organs that support the entire gastrointestinal tract. You could call them the silent partners in human metabolism.
That's a good way to think about them.
We're talking about the salivary glands, the pancreas, the liver, and the gallbladder. And this isn't just a quick look at where these organs are.
No, not at all.
This is uh, more like a microscopic analysis of how they operate, how they chemically engineer their secretions, and really the integrated physiology that defines, well, professional level knowledge in this field.
Exactly. Our sources provide this highly detailed, cellular level roadmap of the histology and function of these crucial accessory organs.
Right.
So our mission today is to synthesize all this complex information. I mean, from the polarity of individual secretory cells all the way to the precise microflows of blood and bile in the liver, to ensure you achieve a really comprehensive, masterful understanding of their functional relevance.
Yeah.
We're definitely looking past the basics here and focusing on the systems that reveal that true sophistication.
And for you, the listener, whether you're maybe preparing for a difficult exam or a clinical briefing, or maybe you're just aiming to be the most deeply informed person analyzing human biology, well, understanding these integrated systems provides incredible perspective on basically everything from digestion to detoxification and metabolic health. Okay, so let's begin the digestive journey right there in the oral cavity with the salivary glands. I think most people think of saliva as just, water.
Right. That's the common perception.
But functionally, it's actually one of the most chemically complex fluids the body produces. So, beyond just moistening food, what are the primary chemical and defense roles of saliva?
Well, functionally, saliva is essential for both mechanical transport and chemical initiation.
No.
Mechanically, it moistens and lubricates the food we ingest, and critically, it protects the oral mucosa itself from drying out or getting damaged.
Okay.
Chemically, it kicks off two major digestive processes right away. Carbohydrate breakdown via something called alpha amylase, and some preliminary lipid hydrolysis via lingual lipase.
And the immune role is pretty significant too, right? It's kind of our first line of defense against a lot of what we put in our mouth.
Oh, absolutely. It is a critical component of our innate immunity. Saliva actively secretes some really powerful antimicrobial agents, notably lysozyme, which actually attacks bacterial cell walls,
Wow.
and lactoferrin, which basically grabs onto the iron that bacteria need to grow. On top of that, saliva maintains a crucial buffering function. It keeps the oral pH pretty consistently between 6.5 and 6.9.
And that protects teeth.
Exactly. Protects dental enamel and the soft tissues, too.
The sheer volume is something that often catches people off guard. We're producing a significant amount every day.
Indeed. The major glands alone secrete somewhere between 0.75 and 1.5 liters of saliva every single day.
A liter and a half. That's a lot.
It is. And then the numerous smaller, minor glands scattered throughout the oral mucosa, they contribute maybe another 10% of that total volume. It's an incredibly high turnover fluid system.
So let's break down the three major pairs of glands because their histological specialization, their structure, directly determines the type of saliva they produce. How do the parotid, submandibular, and sublingual glands sort of differentiate their secretion?
Yeah, they're really defined by the dominance of their specific secretory cells, the parotid glands, the ones situated near the ear.
Okay.
They are purely serous. That means their acini units, the little secretory units, are exclusively made of serous cells. So their saliva is very watery, protein-rich, and it's loaded with that alpha amylase and also proline-rich protective proteins.
Okay, watery and enzyme-rich from the parotid. Then we have the submandibular glands, which you mentioned are kind of the workhorses, producing most of the saliva.
That's right. They're classified as branched tubuloacinar glands, and they're predominantly serous, but, and this is key, they contain mixed units.
Mixed meaning serous and...
Serous mucus cells. So this means they produce alpha amylase, those proline-rich proteins, and lysozyme. But the presence of the mucus cells gives their secretion a slightly thicker consistency than the purely watery parotid saliva. They're responsible for roughly 2/3 of the total daily salivary output.
2/3, okay. And finally, the smallest pair, the sublingual glands.
Right, under the tongue. They follow the same branch tubuloacinar structure, but they are dramatically dominated by mucus cells.
Ah, so mostly mucus then.
Exactly. Consequently, their primary product is thick, viscous mucus. Now, there are a few serous cells present, and they contribute trace amounts of amylase and lysozyme, but their defining product is really protection and lubrication via mucin.
This histological difference, the mix of cells and glands like the submandibular, introduces a structure I remember reading about, the serous demilume. Is this a functional unit or more of a, well, a visual thing in slides?
Ah, the demi-lune. Good point. The term means half moon, and it describes this crescent-shaped cap of serous cells that appears grouped distally on these short mucus tubules in the mixed glands.
Okay.
Now, while they're definitely the site of serous secretion right next to the mucus production, it's generally accepted now that the characteristic crescent shape is, well, at least partly an artifact.
An artifact. How so?
It's created during the slide preparation process. It seems to be due to the differential swelling and fixation of the adjacent hydrophilic mucus cells. Functionally, though, it still represents the dual output of the gland, but structurally it's a specific pattern we look for histologically in those mixed glands.
Interesting. Let's zoom in just a bit on the two main cell types themselves, the serous and mucus cells, because they seem like polar opposites in how they work.
They really are. Serous cells are the classic protein secretors. They're pyramidal in shape, highly polarized, meaning they have distinct top and bottom ends,
Yep.
and they feature a centrally located round nucleus. Critically, their cytoplasm is just packed with rough endoplasmic reticulum, the RER, for extensive protein synthesis. And they store their enzyme products in these large, densely staining apical secretory granules organized into spherical units called acini. They're basically built to rapidly produce and release proteins.
Built for proteins, whereas the mucus cells are built more for, like, viscous hydration.
Correct. Mucus cells are typically more columnar in shape. Their nuclei tend to get compressed and pushed towards the basal membrane just by the sheer volume of their product.
Which is the mucin.
Right, which consists of large apical granules filled with these hydrophilic mucins. Now, because routine slide preparation often involves dehydration, the mucins can sometimes be lost or poorly stained, making the cells look pale or almost empty. And structurally, they tend to organize into cylindrical tubules rather than those spherical acinis we see with serous cells.
And linking these specialized secretory units together, ensuring the product actually gets out, are these vital support cells, the myoepithelial cells.
Ah, yes, the myoepithelial cells. They're kind of the unsung heroes of glandular secretion. They are small, flattened, almost star-shaped cells that are strategically located inside the basal lamina, wrapping around the acini in the initial parts of the ducts.
Inside the basement membrane.
Yes, that's important. And their critical role is contraction. When they get stimulated by autonomic signals, they contract forcefully, squeezing the secretory products out of the delicate acini and into the beginning of the duct system. Without them, that viscous product, especially the mucus, would really struggle to leave the secretory units.
Okay, so the product is made, squeezed out, and here's where, for me, the salivary glands get really interesting. The duct system isn't just passive plumbing, is it? It's like an active ion transporting factory.
Absolutely. This is the refining stage, and it demonstrates immense energy expenditure. The initial product, which is roughly isotonic with plasma, leaves the acini and enters these short intercalated ducts lined by low cuboidal epithelium. But the real action happens immediately as these flow into the specialized striated ducts.
Striated ducts, why are they called that, and why do they need so much energy?
Well, under light microscopy, they show these faint vertical basal striations, which gives them their name. But ultrastructurally, looking closer with an electron microscope, these columnar cells are just masterpieces of active transport. They feature these very deep, extensive infoldings of the basolateral membrane.
The bottom and side membrane.
Right. And perfectly aligned with these folds are just numerous mitochondria.
Okay, and that dense concentration of mitochondria immediately signals high metabolic demand. This isn't just passive diffusion happening.
But anything but passive. The critical defining function of the striated ducts is the active reabsorption of sodium ions, Me+, and chloride ions, Cln, from that initial isotonic primary secretion. This active transport process, powered by the immense ATP generated by all those mitochondria, literally strips electrolytes away from the saliva.
And what's the functional consequence of spending all this energy just to remove electrolytes? Why do that?
The result is that the final saliva product, the stuff released into the oral cavity, is slightly hypotonic relative to blood plasma.
Less concentrated.
Exactly. And why is this critical? Well, hypotonicity is vital for maintaining the hydration and integrity of the oral mucosa without causing major osmotic shifts. And it's also essential for the proper lubrication and dissolving power of the saliva before you swallow. It really demonstrates how the body fine-tunes the fluid profile, not just the chemical content. It's quite elegant.
It really is. So after this active refinement in the striated ducts, the secretion then moves into the larger transport vessels.
Right. These are the excretory ducts, or sometimes called interlobular ducts, which converge and progressively increase in size as they head towards the main opening. Their epithelial lining is actually noted for its variability. It can range from simple cuboidal or columnar to stratified or even pseudostratified.
That variety suggests they're doing more than just transport, too.
Yes, exactly. This structural diversity reflects their varied functions.
Hmm.
You find cells within these unusual epithelia that continue some fine-tuning of ion reabsorption, along with other cells that secrete mucin. You even find some enteroendocrine cells and basal stem cells scattered in there. And importantly, this is where a major local immune defense strategy culminates.
Ah, the IgA defense. How does the duct system facilitate this specific immune transport?
It's a really beautifully coordinated mechanism. You have plasma cells strategically situated in the connective tissue surrounding these excretory ducts.
Okay.
These plasma cells release IgA antibodies. Then, these IgA molecules form a complex with a specific secretory component that's synthesized by the duct cells themselves.
So the duct cells make part of it?
Yes, they make the transport protein, essentially. This whole IgA secretory component complex is then actively transported into the saliva, and that provides targeted, specific defense against pathogens trying to enter via the mouth.
Okay, wrapping up the salivary glands then. The speed and the composition of the secretions seem entirely under the command of the autonomic nervous system.
Precisely. Parasympathetic stimulation, which gets activated by the sight, smell, or taste of food, drives a copious, watery secretion. That's the classic mouth-watering response. High volume, low organic content flow.
Right.
Conversely, sympathetic stimulation, which we associate with stress or anxiety, strongly inhibits this secretion. And that leads to the familiar, sometimes uncomfortable sensation of dry mouth or xerostomia.
And clinically, xerostomia is a serious issue, isn't it? Often caused not just by anxiety, but by actual pathology or medications like the mumps virus, radiation treatment for head and neck cancers, or even common anticholinergic drugs like some antihistamines.
Absolutely. And its opposite, siluria, or excessive saliva production, is often linked to parasympathetic overdrive. That can result from conditions like nausea, sometimes rabies infection, unfortunately, or severe oral inflammation. So that regulatory balance is actually a key indicator of the overall physiological state.
All right, moving down the track now to the pancreas. This is really the heavy hitter in terms of systemic digestion, wouldn't you say?
Oh, absolutely. It's indispensable.
This retroperitoneal organ tucked behind the stomach, it's famous for its dual identity. You've got the endocrine part, the islets of Langerhans making hormones like insulin.
Right, which we'd cover in a different context, perhaps chapter 20 in our source.
Exactly. But today we're focusing exclusively on that massive exocrine machinery, the part that involves these serous acini producing just tons of digestive enzymes.
The exocrine pancreas is a true powerhouse. Structurally, it consists of these serous acini surrounding a very small central lumen. The serous cells themselves are highly polarized and absolutely packed with apical zymogen granules.
Zymogen granules. Those are the storage vesicles for the inactive enzymes, right?
Precisely, inactive precursors.
Okay, so we just talked about the parotid gland, which is also purely serous, and it relied on those striated ducts for ion refinement and the myoepithelial cells for propulsion. How does the pancreas, which is also serous, compensate for the fact that it apparently lacks both of those structures? No striated ducts, no myoepithelial cells.
Yeah, that's a crucial structural differentiator, and it forces a different functional strategy. Since the pancreas lacks myoepithelial cells, propulsion of the secretions relies much more on the pressure generated by the continuous high-volume fluid secretion coming from both the acini and the ducts themselves.
Okay, so just the flow pushes it out.
Pretty much.
Yeah.
And because it lacks striated ducts, it has to achieve its necessary chemical modification, specifically that crucial ion transport right at the very start of its duct system. And it does this using a completely unique cell type found only in the pancreas.
And that unique cell type would be the central acinar cells.
Yes. These are the histological hallmark of the pancreas. You look for these. They are small, pale-staining cuboidal cells that are actually the initial cells of the intercalated ducts, but they physically extend into the lumen of the acinus itself. It's a very characteristic appearance. You only find them here.
And these unique cells are central to what seems like the single most important job of the pancreas, besides making enzymes, generating that massive volume of alkaline fluid.
Precisely. The intercalated ducts, which originate with these centroacinar cells, secrete roughly 1.5 liters of fluid every single day. And that fluid is extraordinarily rich in bicarbonate ions, HCO3 here.
1.5 liters of bicarbonate solution. Why is this high-volume alkaline secretion so necessary? Why can't the enzymes just work in the acidic environment coming from the stomach?
Because the highly acidic chyme pouring into the duodenum from the stomach, we're talking pH 1 or 2, would instantly deactivate or denature those powerful pancreatic digestive enzymes. They just can't function at that low pH.
Okay, makes sense.
So this alkaline pancreatic juice, loaded with bicarbonate, acts as a powerful buffer. It instantly neutralizes the incoming acid and raises the pH in the duodenum to an optimal range, usually somewhere around 7.0 to 8.0. And that environment is absolutely essential for the pancreatic enzymes to function effectively. It basically establishes their proper working conditions.
Got it. Let's detail the enzymes themselves. We know they're secreted as inactive zymogens, which you mentioned is a built-in safety measure.
Right. The full suite of enzymes includes proteases to break down proteins, alpha amylase, like in saliva for carbohydrates, various lipases for fats, and nucleases for DNA and RNA. The proteases, like trypsinogen and chymotrypsinogen, are potentially the most dangerous if activated prematurely. They're synthesized and stored as these inactive zymogens. If they were active inside the pancreas itself, it would lead to, well, autodigestion.
Self-digestion. Not good.
Definitely not good.
So the goal is to keep them inert until they are safely outside the gland and right at the site of action in the duodenum. What mechanism controls that crucial moment of activation?
That is the zymogen activation cascade, and it's very tightly restricted to the duodenal lumen. The entire process hinges on one key enzyme, enteropeptidase.
Enteropeptidase, where does that come from?
It's synthesized by and actually fixed within the brush border membrane of the duodenal mucosal cells themselves, so it's part of the intestinal lining. Enteropeptidase specifically cleaves and activates trypsinogen, converting it into the active enzyme, trypsin.
Okay, so enteropeptidase lights the fuse by making trypsin.
Exactly. And once trypsin is generated, it's like setting off a chain reaction. Trypsin itself is autocatalytic, meaning it rapidly activates even more trypsinogen. And crucially, it also activates all the other protease zymogens, chymotrypsinogen, proelastase, procarboxypeptidase, etc., in a rapid cascade. This unleashes the full digestive power of the pancreatic juice right where it's needed in the duodenum, and not before.
Given the, well, destructive power of these proteases if they go off early, the failure of this system seems catastrophic. You mentioned the pancreas has a kind of 3-layered protection mechanism to prevent this autodigestion. Can we explore those safeguards in detail?
Yes, this is one of the most critical aspects of pancreatic physiology, ensuring its own survival. Safety mechanism. 1 is the physical or mechanical restriction we just discussed, keeping the activation cascade entirely outside the pancreas, confined to the duodenum where enteropeptidase resides.
Okay, location.
Pretty much. Safety mechanism 2 operates right down at the cellular level, inside the pancreas. When the zymogens are packaged into those apical granules in the acinar cells, the pancreas simultaneously co-packages a specific peptide called trypsin inhibitor, right alongside the trypsinogen.
An inhibitor in the same package.
Exactly. So if any minor accidental premature activation of trypsin does occur within the cell or maybe in the duct system, the inhibitor's right there, ready to bind to it and inactivate it instantly. It acts like an internal damage control system.
Clever. And the third mechanism is more functional, tied back to that alkaline fluid environment you mentioned.
Precisely. Safety mechanism 3 is the maintenance of a relatively high pH within the entire duct system, driven by that constant HCO3 or secretion from the centroacinar and duct cells. The pH within the acini and ducts is deliberately kept alkaline, well above the optimal, slightly acidic pH range where these enzymes are most active. So even if the trypsin inhibitor somehow fails, the high alkalinity helps ensure the enzymes remain functionally inert until the pH drops closer to neutral in the duodenum.
Wow, 3 layers of defense. So when all 3 layers fail or are overwhelmed, that's when we see the clinical emergency of acute pancreatitis.
That is the tragic result. It occurs when these proenzymes are activated prematurely within the organ itself. This can be triggered by things like gallstone blockage of the ducts or sometimes severe alcohol toxicity. It leads to uncontrolled digestion of the pancreatic tissue itself. Understanding that triple defense system really highlights why it's so absolutely necessary.
Finally, for the pancreas, let's turn to the hormonal switches that regulate this whole operation. How does the body tell the pancreas, okay, now dump the enzymes versus now dump the neutralizing fluid?
It's very tightly controlled by 2 main hormones released from those enteroendocrine cells located in the lining of the small intestine. The first is cholecystokinin, or CCK.
CCK, what triggers that?
CCK is released primarily in response to the presence of fats and proteins entering the duodenum from the stomach. And CCK specifically targets and stimulates enzyme secretion by pancreatic acinar cells. So if food arrives, CCK is released, pancreas releases enzyme.
Makes sense. What's the second hormone?
The second hormone is secretin. Secretin is released mainly in response to the acidity of the chyme arriving from the stomach, and secretin specifically targets the duct cells, including those important central acinar cells promoting the massive output of water and bicarbonate, H2O3.
Ah, so acid triggers the bicarbonate release.
Exactly. These two hormones work in concert to ensure a perfect combination. The digestive enzymes arrive simultaneously with the necessary alkaline fluid required for them to function properly. It's a beautifully coordinated response.
And just as a crucial medical context point before we leave the pancreas, the duct system, while responsible for this life-saving alkalinity, is unfortunately also the origin site for one of the deadliest forms of cancer.
Indeed, pancreatic cancer tragically has a very high mortality rate. It usually presents as a carcinoma, a cancer arising from the duct cells, and most often it occurs in the head of the pancreas, which is nestled right near the duodenum. Because the organ is retroperitoneal, kind of hidden at the back, and the tumors are often asymptomatic until they're quite advanced, detection is often late. This leads to difficult surgical outcomes and, unfortunately, a poor prognosis in many cases.
Okay, now we move to what you called the master regulator of the entire body, the largest internal organ, the liver. Averaging about 1.5 kilograms, this is the metabolic hub where, fluid mechanics, chemical engineering, and structural organization seem to reach their peak complexity. We absolutely have to start with the foundational concept of its blood supply.
Yes, the liver's dual blood supply is absolutely essential to understanding pretty much everything it does. Only about 25% of the blood flowing into the liver, providing the necessary oxygen supply, comes via the hepatic artery.
Only a quarter for oxygen.
Right. The overwhelming majority, the other 75%, comes from the hepatic portal vein.
And that portal blood is unique, isn't it? It's not typical venous blood.
Not at all. It is nutrient-rich but relatively oxygen-poor. Why? Because it's carrying basically all the absorbed products, nutrients, yes, but also potential toxins, drugs, hormones collected directly from the stomach, the small and large intestines, the pancreas, and the spleen.
Wow, everything drains there first.
Almost everything from the digestive tract. All this blood enters at the hilum, or the porta hepatis, and it absolutely must be processed by the liver before it's allowed to re-enter the general circulation via the hepatic veins. This dual supply, arterial and portal venous, is what fundamentally defines the functional zonation we'll discuss in a bit.
Okay, so this means the main workhorse cells, the hepatocytes, they must handle just an unparalleled diversity of tasks. Let's try and list out their main functional categories. They truly seem like the most versatile cells in the body.
They really are incredibly versatile.
Right.
Functionally, they are responsible for: first, exocrine processes, primarily the production of bile, which is absolutely vital for emulsifying fats in the duodenum, allowing them to be digested and absorbed.
Okay, bile production. What else?
Second, endocrine tasks. They synthesize and secrete nearly all the major plasma proteins into the blood. We're talking albumin, which maintains osmotic pressure,
Right.
fibrinogen and other clotting factors for blood coagulation, epolipoproteins for transporting lipids, transferrin for carrying iron, and many others.
Wow, that's a huge job right there. And then there are the really complex metabolic and storage roles, right?
Absolutely. Metabolically, they are crucial for gluconeogenesis, that's converting amino acids or lactate into glucose when blood sugar is low. They're central to maintaining normal carbohydrate and protein metabolism. They also handle nitrogenous waste by removing amino groups from excess amino acids, converting the resulting ammonia into urea, which is then sent off to the kidneys for excretion.
And the famous detoxification and storage aspects, can't forget those.
Definitely not. Detoxification involves breaking down and conjugating, which basically means chemically modifying to render harmless a huge range of ingested toxins and countless pharmaceutical drugs. It's a massive chemical processing job.
Okay. And storage.
And finally, storage. Hepatocytes store glucose in the form of massive glycogen granules ready for quick release. They store triglycerides in small lipid droplets, and they store iron, complex safely as ferritin. Interestingly, vitamin A and some other fat-soluble vitamins are specifically stored not in the hepatocytes themselves, but in another specialized cell type nearby.
We'll get to that. Okay, let's define the classical structural unit that tries to make sense of all this. The hepatic lobule. How is that organized?
The classic hepatic lobule is typically depicted as a roughly hexagonal or polygonal shape in cross-section. The hepatocytes themselves, which are large, cuboidal, or maybe polyhedral cells, often possessing 2 nuclei and frequently being polyploid, meaning having extra sets of chromosomes.
Okay.
They are organized into these irregular, interconnected cellular plates, maybe 1 or 2 cells thick. These plates are arranged radially, like spokes on a wheel, supported by a very delicate stroma of reticulin fibers, and they're all oriented towards a small collecting vessel right at the geometric center, the central vein.
The central vein is the drain, essentially. And found at the corners or the periphery of this hexagonal structure are the critical inflow and outflow junctions.
Exactly. Those are the portal triads, sometimes called portal tracts or portal areas. You find them embedded within the fibrous connective tissue at the periphery of the lobule, typically at the corners of the hexagon. A triad consists of 3 essential interlobular structures working together. First, a venial branch of the portal vein, bringing that nutrient-rich blood.
Okay.
Second, an arterial branch of the hepatic artery, bringing the oxygenated blood. And third, 1 or 2 small bile ductules, which represent the very beginning of the biliary system, collecting the bile produced by the hepatocytes. So this is where all the incoming traffic arrives and where the bile starts its exit journey.
Got it. So once the blood arrives via the triad, the mix of portal venous and hepatic arterial blood, it enters these low-resistance highways within the liver, the sinusoids, which lead towards that central vein. What's unique about the structure of the hepatic sinusoids themselves?
The sinusoids are irregular, dilated vascular channels that run between the plates of hepatocytes. This is the crucial location where the nutrient-rich portal vein blood actually mixes with the oxygenated arterial blood. Critically, the endothelium lining these channels is very thin, it's discontinuous, meaning there are gaps between the cells, and it's highly fenestrated, riddled with pores or windows.
Fenestrated, like little windows. And unlike typical continuous capillaries, they lack a complete basement membrane, a basal lamina.
Precisely. They're surrounded by only a very sparse, discontinuous basal lamina.
Okay, so this unique architecture creates the perfect sort of leaky exchange site, this area called the space of Dissy. What is the ultimate significance of this microscopic space?
The parasinusoidal space, or the space of Dissy, is the extremely narrow gap that exists between that leaky, fenestrated sinusoidal endothelium and the surface of the hepatocytes themselves. Because the endothelium is discontinuous, blood plasma, not whole blood cells, but the fluid plasma, with all its dissolved contents, freely fills this space, and it directly bathes the dense carpet of microvilli that project from the hepatocyte surface into that space.
Wow, so this means the hepatocyte isn't buffered by a basement membrane or thick connective tissue. It has immediate direct access to the raw materials, the nutrients, the toxins, everything dissolved in the blood plasma.
Exactly. This direct plasma contact is precisely why the liver is such an incredibly efficient regulator and processor. It facilitates the immediate uptake of nutrients like glucose and amino acids, the rapid release of newly synthesized plasma proteins like albumin back into the circulation, and the quick transfer of toxins or drugs from the plasma into the hepatocyte for detoxification.
And the clinical relevance.
It's immense. When liver fibrosis or scarring begins the process leading to cirrhosis, it often first occurs right here in the space of Dissy. Collagen fibers get deposited, thickening this space, disrupting that critical exchange pathway, and severely impeding liver function over time.
Okay, we also have 2 key specialized non-parenchymal cell types, meaning not hepatocytes, that live in or around the sinusoids. Let's start with the liver's resident immune guards.
Right, those would be the Kupffer cells. They are specialized, stellate, star-shaped macrophages that are physically embedded within the lining of the sinusoids themselves. Their primary role is constant surveillance of the blood flowing through.
What do they do specifically?
They phagocytose, meaning they engulf and digest aged or damaged red blood cells, freeing up the heme and iron for recycling. They are also essential for removing any bacteria, cellular debris, or particulate matter that might have slipped through from the portal circulation draining the gut. And they act as potent antigen-presenting cells, helping to initiate immune responses when necessary.
Okay, the cleanup crew and guards. And the other specialized cells, the ones responsible for storing those fat-soluble vitamins you mentioned earlier.
Ah, yes. Those are the hepatic stellate cells, also known historically as Ito cells. They reside in the space of Dissy, that parasinusoidal space. Their defining characteristic under normal conditions is the accumulation of small lipid droplets within their cytoplasm, and these droplets are the primary storage site for vitamin A and other fat-soluble vitamins in the body.
So they're vitamin storage units, but they have another more ominous role.
They do. Functionally, they are normally quiescent, just sitting there storing vitamin A. However, upon liver injury, particularly chronic injury from things like alcohol abuse, viral hepatitis, or metabolic syndrome, these stellate cells become activated. They transform into myofibroblasts.
Myofibroblasts, like
Yeah.
muscle and connective tissue cells.
Exactly. They proliferate, they start synthesizing large amounts of extracellular matrix components, especially collagen. And it's these activated hepatic stellate cells that are the central cellular agent driving the onset and progression of liver fibrosis and ultimately cirrhosis. So they have this dual Jekyll and Hyde personality.
Fascinating and scary. Okay, this entire system with blood flowing from the high pressure, high nutrient portal area towards the lower oxygen central vein creates a very definitive gradient across the liver tissue.
Yeah.
You mentioned this is best visualized by maybe abandoning the simple hexagonal lobule structure and embracing the concept of functional zonation via the hepatic sinus model.
Yes, understanding this functional gradient is perhaps the most advanced concept for really grasping liver function, and especially liver pathology. Since the portal, venous, and hepatic arterial blood mix at the entrance to the sinusoid and then flow continually towards the central vein, there's an unavoidable progressive drop in the concentration of oxygen, nutrients, and even certain hormones across the radius of the classical lobule. The sinus model helps us map function onto this gradient.
Okay, tell us about the metabolic specialization of Zone 1, the area closest to the incoming blood supply, the area of highest oxygen concentration.
Zone I is the periportal zone. It's located nearest the distributing branches of the hepatic arteriole and portal venule within the portal triad. These hepatocytes are therefore exposed to the highest concentrations of oxygen and recently absorbed nutrients.
So they get first crack at everything.
Exactly. Therefore, they specialize in metabolic processes that are highly energy dependent and oxygen demanding. This includes things like aerobic respiration, oxidative phosphorylation, extensive protein synthesis like albumin and clotting factors, cholesterol synthesis, and processes like gluconeogenesis. They're generally the first cells to show regenerative activity after injury, but they're also potentially the first affected by toxins that require metabolic activation, or by bile duct obstruction, which causes bile to back up first in this zone.
Okay, Zone 1 is high energy, high oxygen work. And as the blood flows inward and becomes depleted of oxygen and nutrients, it reaches the opposite end of the gradient, Zone 3.
Right. Zone 3 is the pericentral zone, the region closest to the central vein, which is draining the blood away. These cells receive blood that has the lowest concentration of oxygen and nutrients, having already passed by the Zone 1 and Zone 2 hepatocytes.
So what do they specialize in?
Consequently, they tend to specialize in processes that are less oxygen dependent. This includes things like glycolysis, lipogenesis, the formation of lipids, and importantly, detoxification of many drugs and toxins via the cytochrome P450 system, which is often highly concentrated here.
And this low oxygen environment makes them uniquely vulnerable in certain situations.
Very much so. This low oxygen last-in-line status makes Zone 3 cells the preferential site for fatty accumulation, or steatosis, which is often seen first around the central veins in conditions like fatty liver disease. And critically, they are the first cells to undergo ischemic necrosis cell death due to lack of oxygen if blood flow to the liver is compromised, for instance, in shock or severe heart failure.
Okay, so if a patient has a condition compromising overall circulation, like congestive heart failure, Zone 3 around the central vein is where the damage, the necrosis, is most likely to show up first under the microscope.
Absolutely. Conversely, if a patient is, say, taking a drug that is detoxified primarily by Zone 3 enzymes, then Zone 3 might show specific toxic injury related to that drug's metabolites. Understanding the acinus model and its zones really moves our comprehension from just static anatomy to the dynamic distributed metabolic function of the liver. Zone 2, by the way, is just the intermediate region with transitional properties.
Right. To fully appreciate these functional differences between the zones, we really need to look even deeper inside the hepatocyte itself and understand the specialization of its organelles. You describe them as microfactories dedicated to specific tasks.
That's a great analogy. Let's start with the endoplasmic reticulum, the ER. The rough ER, or RER, characterized by its ribosomes, is incredibly abundant and highly developed, particularly in those Zone I hepatocytes near the portal tracts.
Because they're making proteins for export.
Precisely. Its entire purpose is the synthesis and initial modification and packaging of those major plasma proteins, the albumins, fibrinogen, clotting factors, etc., which are then secreted directionally out of the basal surface of the hepatocyte into the adjacent space of DISI to enter the bloodstream. The sheer volume of RER needed for this massive protein export often makes the cytoplasm in Zone 2 appear slightly more basophilic blue staining, and standard histological preparations.
Okay, RER for protein export. And the smooth ER, the SER, lacking ribosomes, that's the famous detoxification engine, right? What exactly happens within that membrane labyrinth?
The SER is equally abundant and particularly prominent in zone 3 hepatocytes. It is incredibly rich in enzyme systems, most famously the cytochrome P450 family. This is the primary site where biotransformation and detoxification occur.
Biotransformation. That involves multiple steps.
Yes, it's typically a two-phase process. Phase I reactions, often involving oxidation, reduction, or hydrolysis catalyzed by P450 enzymes, usually make the toxin or drug molecules slightly more chemically reactive, often by adding a hydroxyl group. Then, phase 2 reactions involve conjugation attaching another molecule, like glucuronic acid, sulfate, or glutathione, to that reactive intermediate.
And the ultimate goal of this two-phase system is to convert something maybe lipid-soluble and hard to excrete into something the body can actually get rid of.
Precisely. It converts relatively hydrophobic, fat-soluble compounds, which would otherwise accumulate in fatty tissues, into more water-soluble hydrophilic derivatives. These water-soluble conjugates can then be safely flushed out of the body, either by being secreted into the bile or by being released back into the blood for subsequent filtration and excretion by the kidneys.
And there's that interesting point about adaptation.
Yes, a fascinating detail is that prolonged exposure to certain drugs, like barbiturates or toxins like alcohol, can actually induce the proliferation of the SCR within the hepatocytes. The cells literally make more SCR membrane and enzymes, effectively increasing the liver's capacity for detoxification over time. This is the basis for developing tolerance to some substances.
One of the most critical conjugation tasks the SCR handles is dealing with bilirubin, that breakdown product of old red blood cells. It's notoriously problematic if it builds up.
Yes, bilirubin metabolism is a major challenge for the body and a key liver function. Bilirubin is the pigmented, hydrophobic waste product generated primarily from the heme portion of hemoglobin when old red blood cells are broken down by macrophages, like Kupffer cells. If left unconjugated, this hydrophobic bilirubin is toxic, particularly to the brain.
So the hepatocytes have to grab it from the blood.
They do. Hepatocytes must actively take up this hydrophobic bilirubin from the blood, where it's usually bound to albumin for transport.
And what enzyme specifically does the chemical modification, the conjugation inside the SCR?
The crucial conjugation step is handled by a specific enzyme family called UDP glucuronyl transferases, or UGTs. These enzymes are embedded specifically within the SCR membrane. They catalyze the attachment of one or two molecules of glucuronic acid derived from glucose to the bilirubin molecule.
And this makes it.
This converts the toxic hydrophobic bilirubin into water-soluble bilirubin mono and diglucuronide. This conjugated bilirubin is non-toxic and ready for safe transport and excretion, primarily into the bile.
Okay, so this water-soluble conjugated bilirubin is then ready to be pumped out. If this specific conjugation step is defective or immature, what happens clinically? We often hear about this in newborns.
Right. Failure or significant inefficiency of this conjugation process, or problems with the subsequent excretion of the conjugated bilirubin into bile, causes the yellow pigmentation of the skin and eyes known as jaundice or icterus. In newborns, especially premature infants, the SCR enzyme systems, including the UGTs, are often anatomically and enzymatically immature for the first few days or weeks of life.
So they can't keep up.
They can't keep up with the normal breakdown of fetal red blood cells that occurs after birth. As a result, unconjugated bilirubin builds up in the blood, leading to neonatal jaundice or hyperbilirubinemia. If levels get too high, the unconjugated bilirubin can cross the blood-brain barrier and cause neurological damage, a condition called kernicterus. This really underscores the absolute necessity of this specific SCR conjugation function.
Absolutely critical. Besides the crucial ER networks, what are the other major processing organelles prominent in hepatocytes?
Well, the liver is also very rich in peroxisomes. These small organelles are crucial for several metabolic tasks, including the oxidation of very long-chain fatty acids, which mitochondria can't handle well, and importantly, for controlling the toxic byproducts of oxidation, primarily through the action of the enzyme catalase, which breaks down hydrogen peroxide, H2O2, into water and oxygen.
Okay, peroxisomes for specialized oxidation. And storage.
And of course, visible throughout the cytoplasm, especially after a meal, are the large, irregular clumps of glycogen granules, the storage form of glucose. You'll also see small lipid droplets, representing temporary triglyceride storage. The amount of glycogen and lipid can vary dramatically depending on the metabolic state of the individual.
Got it. Let's shift our attention now to the liver's main exocrine function, The formation and secretion of bile. We need to reemphasize that crucial point. Bile flows in the direction opposite to the blood flow within the lobule.
This countercurrent flow is absolutely fundamental to liver architecture and function. Bile synthesis starts within the hepatocytes, and it's secreted across the apical membrane of the hepatocytes, but not just anywhere. It's secreted specifically into the tiniest branches of the biliary tree, the bile canaliculi.
And what's unique about these canaliculi, they aren't like typical ducts, are they?
No, they're quite unusual. Bile canaliculi are not true ducts with their own separate walls made of specialized duct cells. Instead, they are tiny intercellular channels, typically only 0.5 to 1 micrometer in diameter, formed by shallow grooves in the plasma membranes of two adjacent hepatocytes facing each other. The wall of the canaliculus is the apical membrane of the contributing hepatocytes.
Wow, that's tiny. Given that bile contains potent detergent-like bile salts and acids, how is it safely contained within those tiny grooves formed just by hepatocyte membranes?
Containment relies entirely on highly effective intercellular junctions specifically, the tight junctions, zonule occludens, and also adherens junctions and desmosomes that completely seal the apicollateral membranes of the adjacent hepatocytes, just bordering the canaliculus. This molecular seal is absolutely critical to prevent the potentially toxic bile components from leaking out backwards into the intercellular space, the space of DISI, and damaging the surrounding liver tissue or entering the blood inappropriately.
So tight junctions are key. From these tiny canaliculi nestled between hepatocytes, the bile flows outward, towards the portal triads at the periphery, entirely counter to the incoming blood flow. Where does the bile go once it leaves the immediate vicinity of the hepatocyte plates?
The bile canaliculi drain into the next level of the biliary system, which are the bile canals of Herring, sometimes called intrahepatic bile ductules. These do have their own lining, composed of specialized cuboidal epithelial cells called cholangiocytes, mixed perhaps with some hepatocytes initially.
Cholangiocytes, the bile duct lining cells.
Exactly. These canals of Herring then merge quickly within the portal area, the portal triad region, to form the larger interlobular bile ductules, which are lined entirely by cuboidal or low columnar cholangiocytes. These bile ductules within the portal triads are the structures that we identify as the bile duct component of the triad. They eventually converge to form progressively larger ducts, ultimately leading to the right and left hepatic ducts that exit the liver.
Okay, that traces the path out. Finally, before we leave the liver proper, we must mention its remarkable, almost legendary capacity for regeneration. It can apparently lose a substantial portion of its mass and recover entirely. Yes, the regenerative capacity of the liver is truly extraordinary among mammalian organs. Following partial hepatectomy, surgical removal, or acute injury that causes significant hepatocyte loss, the liver triggers a powerful process of compensatory hyperplasia.
Hyperplasia meaning cell division.
Yes. The remaining healthy hepatocytes, which are normally quiescent, not dividing, are stimulated to exit their resting state, G0 phase of the cell cycle, and rapidly undergo mitosis to restore the original liver mass. It's remarkably efficient, which is why living donor liver transplantation is possible.
Is it always the existing mature hepatocytes that proliferate, or is there a stem cell population involved as well?
That's a great question, and an area of active research. While proliferation of existing mature hepatocytes is definitely the main driver of regeneration after most types of acute injury or partial hepatectomy, there is growing evidence suggesting a potential backup system involving resident liver stem cells or progenitor cells, sometimes referred to as oval cells in rodents.
Where are these found?
These putative stem cells seem to reside in or near the canals of Herring, among the cholangiocytes, close to the portal areas. It's thought that in situations of severe chronic liver injury, where hepatocyte proliferation might be impaired, these progenitor cells can become activated and are capable of differentiating into both new hepatocytes and new cholangiocytes, contributing to repair, although perhaps less effectively than hepatocyte hyperplasia alone.
All right, so we've established the flow of bile originating in the canaliculi, moving through the ductules and ducts within the liver. Now let's trace its journey through the larger ducts outside the liver, leading to its storage and concentrating facility, the gallbladder.
Okay, so exiting the liver, the right and left hepatic ducts merge fairly quickly to form the common hepatic duct. This duct then typically joins with the cystic duct, which is the duct leading directly to and from the gallbladder.
Hepatic plus cystic equals.
Hepatic plus cystic equals the common bile duct, and it's the common bile duct that travels down towards the duodenum. From the hepatic ducts onward, the lining of these larger ducts remains generally consistent. It's a simple columnar epithelium composed of those cholangiocytes we mentioned earlier.
And the ultimate fate of the common bile duct is integration with the pancreatic duct, right before entering the intestine.
Yes, usually the common bile duct joins the main pancreatic duct very close to the duodenum, forming a short dilated structure called the hepatopancreatic ampulla, or ampulla of better. The release of this mixed fluid, the bile coming down the common bile duct and the pancreatic juice coming from the pancreatic duct into the second part of the duodenum, is tightly controlled by a muscular sphincter mechanism, the sphincter of Oddi, located right where the ampulla opens onto a little elevation called the major duodenal papilla.
Very precise control there. Now, the gallbladder itself, it's a relatively small, often described as pear-shaped organ tucked under the liver. It apparently only stores about 30 to 50 milliliters of bile at a time. Its main function, however, isn't just passive storage, is it? It's active concentration. How efficient is this concentration process?
It is remarkably efficient. Yes, its primary function is definitely to concentrate the bile by actively reabsorbing water and electrolytes during the periods between meals when bile release isn't needed.
How does its structure support this?
Well, the gallbladder mucosa, the inner lining, is characterized by numerous deep folds when the organ is empty or partially filled. This increases the surface area significantly. This mucosa is lined by tall, simple columnar epithelial cells, which look superficially like intestinal absorptive cells. Crucially, these cells feature prominent microvilli on their apical surface facing the bile and contain massive numbers of mitochondria, especially in their basal region.
Okay, so the microvilli increase surface area, and the mitochondria signal high energy use, structural indicators of intense active transport, just like we saw in the striated ducts of salivary glands.
Exactly the same logic applies.
So we have the structural evidence. How does the active transport mechanism physically extract water from the bile to concentrate it?
The mechanism relies on establishing a powerful osmotic gradient across the epithelium. Specialized sodium pumps, Na plus K plus ATPase, are located specifically in the basolateral membranes of these gallbladder epithelial cells, the membranes facing away from the bile and towards the underlying connective tissue, lamina propria.
Pumping sodium out.
Right. They actively pump sodium ions out of the epithelial cell and into the narrow intercellular spaces and the lamina propria. Chloride ions and bicarbonate often follow. This active removal of solutes from the cell into the tissue creates a strong osmotic drive. Water then passively follows the osmotic gradient, moving from the bile through the epithelial cell, likely via aquaporin channels, and out into the connective tissue, where it's picked up by capillaries.
And the result?
The result is that the gallbladder can reduce the bile volume quite dramatically, concentrating the bile salts, cholesterol, and bilirubin by maybe 5 to 10-fold, sometimes even more.
Wow, up to 10-fold concentration. That degree of concentration definitely sets the stage for potential clinical issues like gallstones. But before we get to that, how is this highly concentrated bile released from the gallbladder into the duodenum exactly when it's needed for fat digestion?
The release is beautifully orchestrated, again, by that hormone we met in the pancreas discussion, cholecystokinin, CCK.
CCK again, released by the intestine.
Yes, CCK is released by those enteroendocrine cells, eye cells, in the mucosa of the duodenum and jejunum, specifically in response to the presence of ingested fats and, to some extent, proteins arriving from the stomach.
And what does CCK do at the gallbladder?
When CCK levels rise in the blood and reach the gallbladder, it binds to receptors on the smooth muscle cells within the gallbladder wall, the muscularis externa. This binding triggers a strong, coordinated contraction of the gallbladder's muscularis layer, which notably features muscle bundles oriented in multiple directions to allow for effective squeezing. Simultaneously, CCK also causes relaxation of that sphincter of Oddi down at the duodenum.
So it squeezes the bag and opens the tap.
Exactly. This contraction forces the stored, highly concentrated bile down the cystic duct into the common bile duct and out into the duodenum, precisely when fats are present and need to be emulsified for digestion and absorption.
Perfect timing. Okay, now the downside. Failure or imbalance in this process of storage, and specifically concentration, can lead to the formation of gallstones or cholelithiasis.
Yes, this is often a direct consequence of that very efficient concentration mechanism. Gallstones typically originate when the bile contains excessive amounts of certain normal components, usually cholesterol or bilirubin, that become supersaturated, especially as water is reabsorbed in the gallbladder. When saturation limits are exceeded, these components can crystallize out of solution and aggregate to form stones.
What are the two major compositional types of stones that we generally see?
The vast majority, maybe 80% or more in Western countries, are cholesterol stones. These result primarily from the bile becoming excessively supersaturated with cholesterol, often due to a combination of factors like high cholesterol secretion by the liver, decreased bile salt synthesis, or gallbladder hypomotility, meaning it doesn't empty well. These stones are typically yellowish-greenish and can vary greatly in size.
Okay, cholesterol stones are most common. What's the other type?
Less common, particularly in Western populations, but more prevalent in some other parts of the world, are pigment stones. These are primarily composed of calcium bilirubinate. They tend to be smaller, darker, often brown or black. Pigment stones usually result from conditions that cause excessive levels of unconjugated bilirubin in the bile.
Unconjugated bilirubin, like from red cell breakdown.
Exactly. Often linked to chronic hemolytic conditions like sickle cell anemia or hereditary spherocytosis, where there's just too much bilirubin byproduct being generated from red cell destruction for the liver to fully conjugate and handle efficiently. Certain bacterial infections in the biliary tract can also contribute by deconjugating bilirubin within the bile ducts.
And regardless of the type, cholesterol or pigment, gallstones cause problems mainly
By leading to obstruction. They can block the cystic duct, causing gallbladder pain, biliary colic, or inflammation, cholecystitis. Or if they pass into the common bile duct, they can cause obstruction there, leading to jaundice, cholangitis, infection of the bile ducts, or even pancreatitis if they block the shared opening into the duodenum. #tag outro.
Okay, what stands out most to you when we reflect back on the entire scope of these incredibly important accessory organs? For me, a consistent theme that really jumps out is the relentless focus on chemical refinement and precise structural compartmentalization.
That's a great observation.
I mean, we saw the salivary glands expend huge amounts of energy via those striated ducts packed with mitochondria just to create a final product that is deliberately hypotonic, fine-tuning the water balance.
And building on that, we mastered the really elaborate three-tiered defense system in the pancreas, which uses specific enzyme inhibitors and that powerful bicarbonate flow to prevent what would otherwise be a catastrophic self-inflicted chemical injury. We connected structure and function by identifying those unique centroacinar cells as the source of that vital alkalinity.
Right. And finally, we came to understand the liver not just as one homogeneous organ, but as this functionally zoned processing center visualized by the hepatic acinis model, where oxygen and nutrient gradients literally dictate cell specialization. This forces zone I hepatocytes to focus on high-energy aerobic protein synthesis, while zone 3 cells specialize in lower oxygen processes like detoxification, making them uniquely vulnerable to blood flow issues or certain toxins.
Absolutely. And if we connect all this to the bigger picture, I think we see that sophisticated physiological function really requires this constant interplay and counterregulation. Think about the flow of blood moving in the exact opposite direction to the flow of bile within the liver lobule.
Yeah, the countercurrent flow.
Or the hormonal switches CCK versus secretin that precisely balance enzyme versus bicarbonate output from the pancreas based on the duodenal contents.
Right.
Or even the active ion transport that concentrates bile tenfold in the gallbladder. These organs are not simply accessory components dumping products. They are highly specialized chemical and flow engineers, absolutely essential for metabolic survival and homeostasis.
Beautifully put. Okay. One final provocative thought for our listeners to perhaps explore further. We noted that the liver possesses this incredible, almost unique capacity for regeneration through compensatory hyperplasia of existing hepatocytes. Yet when faced with chronic injury, the very same liver can succumb to devastating, often irreversible fibrosis and cirrhosis, driven primarily by those activated hepatic stellate cells producing scar tissue.
Right, the Jekyll and Hyde cells.
Exactly. So the question is, how does the body distinguish between the necessary signals that trigger beneficial regenerative growth, which maintains the liver's delicate architecture, and the overwhelming pathological signals that cause those stellate cells to transform and relentlessly produce scar tissue that ultimately destroys function? Is this distinction solely a matter of the intensity or duration of the injury signal, or perhaps which specific cell type, the hepatocyte needing replacement versus the stellate cell sensing damage, receives the dominant overriding signal to proliferate and act in response to sustained damage? Something to mull over as you continue your deep dive.